So, again, thank you very much for inviting me. Actually, my hero is John Walsh. He's talked me into more things than that I can imagine, and actually pushed me in ways that have been great for my career, for my family, and to make me personally happy. I think that David and I are being focused, but in this room are a whole bunch of researchers, and I think people should just stand up, the researchers stand up for just a moment. And to say, I know they don't want to stand up, but raise your hand at least. Okay. Okay. We don't work independently. We share tissues, we share all sorts of information. We come to these meetings, and we've done it for nearly 3 decades now of coming together to basically defeat alpha-antitrypsin deficiency and to bring therapies to you. And I think that these individuals become some of my best friends and people that are very close to us, and it's always a pleasure to do it. But always in the background, I think, is the need. Many of us are clinicians. We take care of alpha-antitrypsin deficient individuals virtually every day, and we bring home every time the message that we need new and better therapies for alpha-antitrypsin deficient individuals. So I've been charged with asking another question, which is, can we fix liver disease with gene therapy? I've been involved in gene therapy for a long time. In fact, I started in Ron Crystal's laboratory in about 1986. One thing about science that you have to remember is an idea can come, but you need the technologies to basically make those more mature. So when David discovered polymerization, concepts about what kind of things could treat were they were almost not on the table at that point. So as we gain more technologies, we're able to develop better therapies and to hone in on these dreams that we have of having specific therapies to work. And that's been very true also in gene therapy as well. So let me start with, there are approaches to gene therapy that I'd like to talk about specifically targeted to lung disease, but we can also talk about as well liver disease as well. So the one that I'm most familiar with and the one that I've worked with very closely with one of my collaborators, Terry Flott, is muscle-delivered alpha-antitrypsin. The concept being that the muscle will become a factory for making alpha-antitrypsin. Other methods are delivery directly to the liver and also systemic therapy, where we can take a virus that can actually hone in to a particular area, and we have serotypes of some of these gene therapy vectors, which actually are very specific for different tissues. We can look at airway delivery, and there are some challenges to that. And then finally, there's pleural delivery, and my understanding is from Ron Crystal that they are about to start a therapeutic trial in human gene therapy and pleural within the next 6 months in New York City. So let's start with some of the issues and some of the ways in which we approach this problem from that standpoint. There are a number of different types of vectors we can use. And so the first question you would ask is, why do gene therapists focus on viruses? You have to think about it for a moment. Viruses are professional gene deliverers. They have evolved over time to basically evade our immune systems and to deposit their DNA or RNA into our cellular systems. They co-opt our cellular systems and allow for replication of proteins that are encoded in the viruses. And so viruses are a great place to start as far as gene therapy vectors, to take a therapeutic gene and to put it into target cells. There are several different types. Retroviruses is one of the first ones that we've used. We used that back in the 1980s. The first gene therapy projects that were done were in the bubble boy children that were the deficiency, the ADA deficiency individuals, and there was remarkable promise originally starting with those individuals. Unfortunately, the original retroviruses unfortunately had what we call oncogenes, which cause basically tumors in these particular children. And we've always been mindful of that problem as we look for better vehicles to transfer normal genes into people. Adenoviruses was another virus that we've used, and adenovirus is really great at delivering huge amounts of targeted genes to virtually any tissue. The problem is they invoke a powerful immune response, and except in tumors, adenoviruses are not used, and particularly not in genetic diseases at the present time. Adeno-associated viruses developed at the University of Florida were discovered in co-purification of adenoviruses. So they were located in there. And one of the things that was important about adenoviruses is that they don't cause any known pathology in contrast to adenoviruses. Adeno-associated viruses are not associated with any disease. It's a parvovirus. And it has, but it has a limitation, and that is that because it's small, it can't carry large genes in it. So putting the cystic fibrosis gene in is a real trick to put it in, to use it as a target from that standpoint. There are ways in which we can package just DNA into lipid-like molecules that basically merge with the cell membrane, and we can actually take naked DNA as well. And in fact, using naked DNA, you can coat gold particles and do what we call a gene gun and inject it into people from that standpoint. Most of the field has been moving, particularly for genetic diseases, has moved towards adeno-associated viruses. Now, adeno-associated viruses are basically particles. This is an idea of, this is a picture of adeno-associated. This is a serotype called AAV2, which are some of the serotypes that we've used in our human gene therapy studies. You can see the size difference between there's an adenovirus, and there is one of the adeno-associated virus. It's, again, as I mentioned, it's a parvovirus. It's a very small genome, 4.7 kb. Well, how big is alpha-1 antitrypsin therapeutic gene? It's approximately 1.2 kb. So we can definitely fit that into this particular thing. It's an icosahedral thing. It requires a helpovirus, but it is non-pathogenic. And that's one of the most important things about using this wonderful vehicle to transfer genes into. The life cycle of adeno-associated virus is basically it's helped by adenovirus. We can isolate these genomes here, and then we can have basically another cell line, one who has information for packaging only, no ability to replicate. And so that's one of the key concepts about viral-induced gene therapy, is it has to be a dead end. We don't want a situation where when we put a therapeutic gene in, it just keeps replicating and replicating and replicating. So we basically become a total factory for making alpha-1 antitrypsin with no way to turn it off. So these are basically they're non-replicating vectors from that standpoint. And so that's very, very important from gene therapy. So this is how we create these vectors. We basically can take this is the wild type right here. The rep and cap are important for replication of the protein. We can take these guts out and then put in our therapeutic gene along with a promoter. That is information required to make the gene transcript work. And then we can put it into a separate packaging cell type that will allow for donating this information, but it won't be incorporated into the final package. Again, the final package will just have this coated in a viral particle. So recombinant alpha-1 AAV2, its package limit is about 4.7 kilobases with the ITR protein. We have been using a CMV enhanced chicken beta-actin promoter, which basically produces a lot of alpha-1 antitrypsin in different subtypes, and it has a very efficient gene transfer. In fact, the gene transfer is quite high, and we can not only make the transfer very, very high, but we can also make it very, very specific. Different serotypes or target to different areas. Like, for instance, AAV9, it targets to basically neurotissue. AAV2 targets mostly to muscle tissue. AAV8 targets to liver tissue. So we have all these little tools we can pick off and do our therapeutic gene of interest in this case. And you can see here, you can localize this. This can also localize into the chromosomes as well, but it only infects terminally differentiated cells, including hepatocytes and myofibers. So we can now, there are examples in the literature of basically AAV going into virtually every single tissue to be used. So the way we do it is basically we create a vector, we deliver it to a muscle cell by injection, and then that muscle cell can become a factory for making human alpha-1 antitrypsin. And that's the basis of the concept. I'm going to just briefly talk, again, first starting, we take this methodology using mice. Dr. Song was able to inject it into the hindquarters of mice, and you can see these large jumps in the amount of alpha-1 antitrypsin that are produced in the circulation. We knew that if we could do that in humans, that would be wonderful. And so we did a series of studies. And we started with first a series of a small cohort of 9 individuals and 3 different dosing groups. And we would inject up to 9 injections of the total virus load into the deltoid muscle of an individual. This is a picture of ultrasound showing when we're getting ready to inject it into the area. You can see like this. This is actually a blood vessel. Right here, we don't want it injected directly into the blood vessel. We want to inject it into a muscle bundle. So we aim a little bit differently like that. And you can see actually the needle going in. That hard site right there. The needle is coming right down like that. And you can see, and that's the injection site sort of ballooning up the muscle fibers between there from that standpoint. We did that in a series of experiments, and we've done both a combination of 9, we've done 18, and we've also done a huge number, about 180 injections in individuals as well from that standpoint. The summary of the data is that we were able to show that it's safe in alpha-1 antitrypsin deficient individuals, but only small amounts of alpha-1 antitrypsin have been made. We can detect that alpha-1 antitrypsin for more than a year. But we're at the point where we need to be able to increase the amount of of exposure of cells that we need to do. And so right now, Terry and his group are basically doing what's called a clamp type of experiment, where they actually put a cuff around the leg, inject large amounts of the viral vector into the leg. It stays there, and it affects the whole leg completely from that standpoint. And we're waiting for the results of that particular study at the present time. So it's been a long time in coming. There have been 3 major trials that have done human alpha-1 antitrypsin gene therapy at the present time. The answer is yes, we will have human gene therapy for alpha-1 antitrypsin deficiency, for the lung disease, I hope perhaps for the liver disease as well in the future. And the time, I would say, is exactly what David said. Approximately 10 years, hopefully some of these people will not remove. But really, we're getting very close. When we're in clinical trials, it's just a matter of knocking away the challenges that come up at each of these points. And so gene therapy is probably a lot closer than we may think in this particular case. Thank you very much. Questions?
Why targeting muscles?
Why do it in muscles? That's a great question. The main reason to do it in muscle is it's easily accessible to deliver it. I think it's the primary thing. There are some drawbacks to muscle, though. And one of them is that muscle is not a professional secretory cell. And so as we think about other targets, definitely one place that we might want to think about is going back to the liver, which normally makes alpha-1 antitrypsin, and injecting into that area. That's a much more invasive approach, though, having to inject into the liver. But I think it's possible to do, particularly with some of these transjugular approaches that we can get right to the liver from that standpoint. But it's a great question.
Mark, but you mentioned that you can do systemic application of the material. So would that be a route to the liver?
Well, it hasn't been tried in humans yet, but we have done it in mice systemically, and particularly because we have serotypes that home in on specific tissues, it may be possible to do that. There are some concerns about doing systemic delivery. One of the key things that we think about, particularly from an ethics standpoint, is that if you do it systemically, it's possible that it'll locate in the gonads. And under those circumstances, we can alter the gene, basically future generations. This is a no-no, and so we're very careful about that. And in fact, as we do all of our gene therapy experiments, we always measure the amount of our target gene that ends up in other places, particularly in the sperm.
Okay, thank you, Mark. We really appreciate it. Very stimulating discussion and presentation, and a lot of hope here. And I don't know about anybody else, but I'm going to hang around another 10 years to get that cure. How about you? But we're all...